Hello and welcome to my presentation entitled Sun and Symbionts, Translocation in Caribbean Corals. My name is Jesse Crandall and I'm a PhD candidate at SUNY ESF in Syracuse, New York. This is my capstone presentation for my PhD in chemistry. I'd like to begin by thanking a number of people that are instrumental in my PhD journey. First of all, my major professor and advisor, Mark Teese. Dr. Teese has been a great mentor and given me great advice throughout my time at ESF. I'd also like to thank my steering committee, Dr. Mary Alice Koffroth and Dr. Dave Keeper, for their advice along this process. My examination committee of Dr. Krista Murrah and Dr. Gordon Patterson from ESF, and the chair of the examination committee, Dr. Richard Smarden from ESF, will play a role, and I'd like to thank them as well. To begin with, I'd like to give you an overview of what I'm going to present to you today. My research is centered around coral reefs and the role that they play in the marine environment. I've studied coral reefs, and I will explain to you what exactly coral reefs are and how they function in marine ecosystems. My research involves the symbiosis between corals and their symbionts. These symbionts are called Symbiodinium, and I'll go into what that relationship entails and how that symbiosis is important to the corals, the coral reefs, and the marine environments. The research that I do that looks at symbiosis tracks the nutrient acquisition of corals. And so that is the research that I will present to you, is how I have used various chemical techniques and other analyses in order to track nutrient acquisition. And then finally I'll talk about the implications that my research has for the future of coral reefs and for the future of marine environments. So first of all, I want to build a case for you on the importance of coral reef research and the impact that coral reefs have on the marine environment as well as the global environment. So thousands of species of fish and marine organisms are associated with coral reefs. So they're a biodiversity hotspot. Many fish species spend part of their life cycle, whether it's being born on reefs or growing up on reefs or living on reefs in these areas. And countless other tiny organisms live on reefs and are primary producers and part of the food chain that makes coral reefs very productive and very diverse. In addition to this biodiversity, coral reefs serve as a major food source for many people around the world. About 50 million people use coral reefs as their primary source of fish, which is as a source of fish, which is their primary source of protein. There's major commercial fisheries that take place on coral reefs, and that's a major economic driver around coral reef environments and the communities that live near these reefs. And then, as as many of you have probably experienced, coral reefs are a major tourism income source in the tropics. People go to the tropical coral reefs to snorkel, to scuba dive, and just to sightsee because these reefs are beautiful to look at. So they have a global economic impact of somewhere in the range of $30 billion per year, and the estimates go up from there. In addition to the biodiversity, the fisheries, and the tourism, coral reefs also provide protection to the shorelines around them. So as you've heard, they're often referred to as barrier reefs, and this means that they serve as a barrier between weather events, big waves, or hurricanes, and the shores that they protect. So for all of these reasons, coral are very important. And so thriving coral goes hand in hand with a thriving ocean. In order to get all of these benefits from coral reefs, they have to be healthy and productive. And all of these benefits will lead to a thriving ocean. We are in a time of changing oceans, and we don't want necessarily the changing oceans to lead to changing and dying reefs. What we do want is to maintain productive reefs. Here is an example of a reef that has changed due to climate change in the Florida Keys. And in the foreground, there's lots of dead staghorn coral, and in the background is another mounding species of coral that has also died as a result of changing ocean conditions. So we want to study coral reefs to understand what types of corals are going to be resilient in a changing ocean, and to understand how corals might change with the changing ocean, in the hope that we can preserve and protect coral reefs for future generations. So these changes that are going on in the ocean are a result of changes that are going on in the global environment. The burning of fossil fuels since industrialization has increased carbon dioxide levels in the atmosphere greatly. And this increased carbon dioxide causes more carbon dioxide also to dissolve into the oceans. This in turn leads to ocean acidification. So the carbon dioxide dissolving into the ocean lowers the pH of the water, thus making the ocean more acidic. This is of critical importance to coral reefs because when corals grow and the way that they form this barrier or this structure for habitat or this barrier to storms is by laying down a calcium carbonate skeleton. Calcium carbonate will dissolve if it's put in strong acid. So as the ocean becomes more acidic, it costs more and more energy for the coral reefs to make this calcium carbonate skeleton. So ocean acidification can lead to a decline in reefs. In addition, rising sea temperatures also harm coral reefs. So we've seen sea surface temperatures rise over the past 75 years or so, and this has a detrimental effect on coral reefs because it impacts how corals grow, and it impacts coral symbiosis. Warmer temperatures mean that different energy systems get used, and different energy acquisition systems need to be used also. So all of these stresses have led to a decline in coral reefs. In addition to this, they've been subjected to overfishing, so poor management. There's been disease outbreaks that have killed lots of corals, and there's also pollution. Now, whether this pollution comes from agriculture or storm runoff or point source discharges, all of these have a negative impact on corals. So I've started to talk about some of the impacts on corals and some of the changes that have taken place in their habitats in recent times. And now I'd like to talk about the coral itself and how coral actually functions. So coral is an animal, and it forms colonies, and each colony is made up of thousands and thousands of small animals. We call these individuals polyps. These polyps have tentacles, and they can engage in heterotrophic feeding. So this is one of the ways that a coral can get its nutrition. The other way that a coral can get its nutrition is from its symbionts. Within the coral animal, there are symbionts called Symbiodinium, and these Symbiodinium are dinoflagellates, which is a single-celled algae, and they're photosynthetic organisms. These photosynthetic organisms use the sun and use the nutrients in the water in order to produce energy and to produce nutrients. They can then give these nutrients to the coral through a process called translocation. So the coral animal can get its nutrition from feeding on particles or plankton in the water, and it can also get its nutrition by simply bellying up to the buffet that its symbionts provide. This symbiosis is absolutely critical to coral health. If you've ever heard of coral bleaching, this is when corals lose their symbionts. When corals lose their symbionts, they no longer have the ability to get nutrients from them. And if they don't get new symbionts, they will not have enough energy and they will die. In shallow surface waters, in shallow environments, shallow habitats, corals are able to get all of their nutrients from this translocation from photosynthetic symbionts. So at the surface, as the figure shows, almost all of the nutrients come from photosynthesis and only a small amount come from feeding. As corals grow in deeper waters, they typically would transition to more heterotrophic feeding because light gets scarce the deeper you go. So the deeper the coral goes, the more it will rely on that heterotrophic feeding on the plankton in the water, and only a little bit on photosynthesis because there's less light. So the focus of my research is to look at symbiosis using chemical techniques and biomarker molecules in order to understand where nutrients are coming from in various corals that live on the reefs, and try to understand why some corals are healthier and can live in a variety of habitats. So, nutrients. Corals need nutrients to survive, and they can get these nutrients from their algae or they can get them from feeding. If they aren't able to get the nutrients from the algae, hopefully they can get them from feeding. But generally there's a mix between feeding and translocation. And understanding this mix will help us to preserve and restore coral reefs so that they can maintain their productivity. So the first piece of research that I'm going to share with you today is a nutrient study that I performed in the Florida Keys. Typically corals grow in nitrogen-limited waters, so I wanted to look at the nitrogen sources that corals get. Corals can assimilate nitrogen in the form of nitrate, ammonium, and urea. And these three nutrients in nutrient-limited waters can be impacted by pollution. It could be agricultural runoff, could be sewage runoff, or it could be storm runoff. The nutrient that I wanted to look at was urea, because urea, while there is a low-level background concentration in marine waters, and there are some producers in coral reef environments as well, urea can be a biomarker for human pollution because sewage contains high levels of urea. The production of urea in reef environments can come from fish pee or also from benthic organisms, which are the organisms that live in the bottom sediments of the ocean, and also from seagrasses. So the picture on this slide is a picture of me sampling out of a benthic flux chamber, and this chamber isolates a small section of the sediments and it collects all of the production in that isolated chamber over a period of time. And then I sample the water that's in that chamber that has the whatever production has happened, and then we analyze that. And we analyzed it for urea concentration. So to look at this urea nutrient, so in looking at this urea nutrient, I wanted to evaluate whether there was pollution from human sources on coral reefs. And what I found was that in nearshore environments, so in these samples that I took from a canal near Key Largo, there was a variable and high level of urea in these waters. This could be attributed to leaky septic systems, to runoff from, and runoff most likely. However, as soon as we got a few kilometers from shore, 3 or 4 kilometers from shore, those high levels dropped right down to a consistent background level of urea. And as we tested urea out to about 13 kilometers from shore, we saw the same consistent background in all of the samples that we collected. There are some producers of urea, as I mentioned, schooling fish and fish pea and seagrass beds, and there were certain times when there were higher concentrations of urea produced in these areas, but there was no accumulation of urea. And so there was no evidence of any human urea signal beyond 4 kilometers from shore. So what this tells me is that the nutrients that a coral reef is able to acquire isn't, when we get further than about 4 kilometers from shore, these nutrients aren't impacted by anthropogenic pollution. In addition to nitrogen nutrients, I wanted to use a technique called metabolomics to start to look at more types of nutrients and to look at the symbiosis between a coral and its symbiont. Metabolomics is a technique that has grown out of the omics family that started with genomics, which looked at the entire genome of a species, so evaluated all of a species' genes, or all of an organism's genes. And then techniques to look at all of an organism's proteins, or proteomics, was developed. Finally, now we're in the age of metabolomics, which looks at all of the metabolites of a species. There's a society for metabolomics, and there's also a metabolomics journal. So there's many plant and animal applications, but not very many marine applications because of challenges that are associated with actually dealing with marine samples. First of all, you have to actually sample the organisms in their environment, and then you have to deal with the salt water, which can be an interference. And so the method that I developed for coral metabolomics, I dealt with these issues, and I was able to analyze the 2 symbiotic partners, the coral animal and the Symbiodinium. I was able to analyze those separately and look at their metabolites. And metabolomics gives us so I used this to study nutrient acquisition, and I looked at the 2 symbiotic partners separately, as I mentioned. I wanted to target biomarker molecules, and so the metabolites that I was able to detect included amino acids, which are used for cell structure and proteins, fatty acids and sugars, including disaccharides, that are energy molecules and used for metabolism, and then finally sterols, which are essential molecules for membrane function. These sterols turn out to be very good biomarkers, and I'll talk a little bit more about those as we go along. So metabolomics is a snapshot of an organism's metabolism. And it analyzes the free metabolites. These metabolites that we analyze in this technique are, as I said, free metabolites, and this means that they're the metabolites that can participate in active metabolism and active translocation from symbiont to host. What we found was that symbiosis is both dynamic and rapid. So it's a variable process, and what appears to be likely is that the symbiont produces metabolites, and very quickly the host incorporates them, or they're translocated and incorporated into the host tissues. One of the metabolites that we were able to detect through this technique is a biomarker called gorgosterol. And you'll hear more about gorgosterol as I continue to present today. But gorgosterol is a biomarker for translocation. It's a sterol that's produced only by the coral symbionts. So from coral metabolomics, from metabolomics of hard corals, I wanted to look at another species of coral that's abundant in tropical reefs, and I chose to look at soft corals. And I chose to look at a variety of species of soft corals. These soft corals are also called octocorals, and I looked at 4 different species across the euphotic zone, so a limited depth range where there is abundant light, and I used biomarkers and other chemical properties to study their nutrient acquisition strategies. In these four morphologically variable soft corals, I found that their nutrient acquisition strategy was essentially the same, regardless of species and regardless of their habitat across the limited depth range that I studied. I looked at biomarker sterols in particular, and I was able to identify 13 sterols that were common to all of the samples. These sterols included biomarkers for feeding as well as biomarkers for translocation. And this translocation biomarker is the gergosterol that I mentioned earlier. And so the depth range that I looked at the depth range that I looked at would still be considered shallow, productive, and abundant light reefs. However, all of the same sterols were present in all four of these species, and they were all present in similar abundances as well, which is evidence for feeding and for translocation all throughout this depth range. So from studying these octocorals, or soft corals, these four diverse species also contain diverse symbionts. So as I mentioned before, Symbiodinium is a diverse group of dinoflagellates, many, many species, and these species can vary by the species of coral that they live. The species of symbiont can vary by species of coral and by habitat conditions. So depending on whether there's high light or low light or temperature considerations or nutrient considerations, all of these things will impact which type of Symbiodinium actually inhabits these corals. The octocorals all had a common sterol composition, and they all contained biomarkers for both feeding and for translocation. So despite having different morphologies and different symbionts, they all had the same nutrient acquisition strategies. Now the octocorals that I sampled them in fairly shallow environments, but they, along with many other corals, can inhabit reefs over a wide depth range. So the next piece of research that I'm going to present involves research on mesophotic reefs. Mesophotic reefs are deeper reefs, and these reefs may be able to repopulate shallow reefs after mortality events. So some of those stressors that I mentioned earlier, such as coral bleaching or storm events or pollution events that can cause corals to be killed, some of that may be mitigated when a species that can survive in both deep water and shallow water can repopulate from deep to shallow. This idea is known as the deep reef refugia hypothesis. And this hypothesis simply states that species can reproduce upward through the depths and repopulate a shallow reef. So I set about to look at a species that lives in both deep habitats and shallow habitats. These deep habitats, or the mesophotic zone where light is very scarce, starts at about 30 meters and extends down to about 150 meters where photosynthesis can't take place anymore. But in this range, in this mesophotic range, there's 1% or less of surface light. So it's pretty dark. I don't have any pictures of sampling down there because the only pictures that are taken are done with expensive dive lights. However, photosynthesis can still occur. So specialized symbionts are able to undertake photosynthesis and possibly provide nutrients to their coral hosts. So what do we expect to see happen at depth? Well, this is the same thing that I talked about earlier. Conventional wisdom says that shallow corals get almost all of their nutrients from photosynthesis. When light is abundant, corals rely on these translocated nutrients in order to get all of their energy needs. They only rely a little bit on feeding. However, in deep reefs, when light is scarce, corals can transition to feeding on the zooplankton and phytoplankton and particulate organic matter that's present in the water column, and not rely on photosynthesis because light is scarce. The evidence for this comes from stable isotope analysis of samples, and I'll talk more about how I use stable isotope analysis, as well as culture studies where light is controlled and feed sources are controlled. And in these cases, we see this, researchers have seen this behavior. So I wanted to look at, as I said, I wanted to look at a mesophotic species and look at it growing in shallow environments as well as deep environments, and look at its nutrient acquisition using chemical analysis of some of these biomarkers. So Montastraea cavernosa grows in both shallow reefs and in mesophotic reefs, and it grows quite well. I did this work in collaboration with a group in the Cayman Islands, so they were able to go down and collect samples at approximately 200 feet below the sea surface, so that I was able to analyze them. We also collected samples that were growing at about 60 feet. So with these samples that came from shallow reefs and mesophotic reefs in the Cayman Islands, I looked at an analysis of biomarker sterols. And the two sterols that I looked at were gorgosterol, which is that sterol that is a biomarker for translocation from the symbionts, as well as cholesterol, which is a biomarker for feeding. Cholesterol is the most abundant sterol in the plankton that coral most often feeds on. By analyzing the abundance of these biomarkers, I could see whether or not these sterols were present and actually available for translocation to the coral host. And then by using stable isotope analysis, I could look at the stable isotope signature of these biomarkers and see how they were not only understand more about their source, but also understand how they were impacting the overall composition of the coral. And to do this, I used a technique called compound-specific stable isotope analysis, and this enables me to separate out each of these sterol biomarkers and then analyze the isotope of these individual biomarkers. So first, I want to tell you about the abundance of these sterols in Montastraea cavernosa. The biomarker for translocation, gorgosterol, is seen in detectable amounts in both the shallow colonies and the deep colonies. So the shallow colonies, once again, are the colonies that were at about 60 feet. The deep colonies were the colonies that were in the mesophotic zone at about 200 feet. So that's a biomarker for translocation that I'm seeing in both habitats. The biomarker for feeding, cholesterol, is also seen in both habitats. So this tells us that there is translocation and feeding going on in shallow habitats and in deep habitats. These are just 2 of the 10 sterols that I detected. And among those 10 sterols were other biomarkers and then sterols that can be acquired by both feeding and translocation. So I went further with these 2 biomarker sterols and all of the other sterols that I detected to look at the stable isotope value of each of these. The reason that I wanted to look at the stable isotope value is because it tells me about the source and then also how it's incorporated into the coral. So when an organism incorporates a nutrient, its stable carbon isotope value takes on the value of that nutrient. And so the carbon isotope value for the bulk of an organism is basically a weighted average of all of the isotopes that is consumed. So if we can identify what the sources are and look at those isotope values, and then we can look at the isotope values of the bulk, we can see where the bulk, what the bulk is made of, and where these nutrients are coming from. So it's a nice way to track what an organism essentially has been eating, or what it's been getting via translocation. So the stable isotope values associated with gorgosterol in this case, the biomarker for translocation, the stable isotope value decreases quite a bit when we go from the shallow samples to the deep samples. This is to be expected because as photosynthesis is taking place in these deeper, low light environments, it takes place much more slowly. And this slowing of photosynthesis causes the carbon isotope value of gorgosterol to become depleted. And this is something that's seen in many different organisms and many different nutrients. So these photosynthetic nutrients that are produced by the symbionts have exactly what we would expect to see as far as their stable isotope value. In the shallow waters, their value is approximately minus 16 per mil, and that's how we measure what's called the delta carbon 13, which is the carbon isotope value. And then when we get into the deeper waters, it's depleted down to minus 24 per mil. In the cholesterol that comes from feeding, the stable isotope value is essentially is pretty similar between the shallow and the deep samples. And this is because the coral is consuming this phytoplankton or zooplankton or particulate matter in the water column, and the cholesterol is going to have a similar stable isotope value because it comes from a similar source. So when we look at the stable isotope value of the bulk, what we see is that the bulk from shallow to deep becomes depleted. And this is just further evidence for the fact that translocation is taking place in these corals. So the shallow samples more closely resemble the stable isotope values of the nutrients acquired in the shallow waters. And then as we go deep, we see that the bulk stable isotope value is depleted because it is still getting these biomarkers. It's still incorporating these photosynthetic nutrients that have a depleted stable isotope value. So this depletion indicates that the coral is still acquiring nutrients by photosynthesis, even at depth. So photosynthesis slows, the stable isotope value of these photosynthetic nutrients decreases, which in turn causes the stable isotope value of the organism as a whole to decrease. So let's go back to what I mentioned earlier about the conventional wisdom. Montastraea cavernosa across depth does not transition to feeding for all of those reasons that I've just talked about. We expected to see a high signal from photosynthetic biomarkers in the surface or the shallow samples and not much signal from heterotrophic feeding. And then as we went deeper, we would expect to see a transition to feeding, so a high signal from the heterotrophic biomarkers and not much signal from the photosynthetic or translocation biomarkers. However, what we did see was that there was no transition to feeding, and the nutrient acquisition was similar in the shallow samples as well as in the deep samples. So those biomarkers for translocation, or that biomarker gorgosterol for translocation, was found in both the shallow and the deep samples in similar abundances. And the bulk stable isotope value of the deep sample reflected the influence of those translocated nutrients. So what this is saying is that in low light, translocation remains important to these corals. So what does this say about Montastraea cavernosa being a refuge species? Well, if it operates in a similar way in the shallow conditions as well as in the deep habitats, it would follow that it can repopulate from deep up to shallow because its nutrient acquisition is essentially the same at both depths. Because it can maintain this, it has a mix of translocation and feeding, so it would potentially be a very good refuge species. So finally, This brings us to the implications for this research. I presented some new analysis tools for investigating symbiosis. Metabolomics is a powerful tool that can be used to better understand translocation between coral symbiont and coral host. And it's shown us that it's identified a number of biomarkers for translocation. I used these biomarkers as well as stable isotope analysis to show that corals acquire photosynthetic nutrients even when light is scarce. And the refuge species that I've talked about can acquire nutrients in a similar way even when light is very scarce. So regardless of what depth it's living at, it gets nutrients from feeding and from translocation, which should make it a resilient species. And diverse refuge species can keep reefs productive, even when ocean conditions change. So using all of these tools and techniques, we can work to identify lots of different species that can be emphasized when coral preservation and coral restoration efforts are taking place, which is all in the goal of ensuring that we have thriving coral, which goes hand in hand with a thriving ocean. And with that, I'd like to thank a number of other people, including my major professor, Mark Thies, again. Without him, none of this would have been possible. Because when I started this journey about 5.5 years ago, I came to ESF and I met with Dr. Thies, and he got me excited about coral reef research. So without him, I wouldn't be where I am today. So thank you very much. Another person at ESF that's had a huge impact on me is Dr. Kelly Donaghy. She has been my boss, my teaching mentor, and my friend throughout my time here. And her advice, her counsel has been absolutely integral to my experience here. My collaborators played a huge role in all the research that I've done. Dr. Mary Alice Kaufroth has been a great mentor to me, as well as getting me started down this path of looking at metabolomics. Dr. Howard Lasker helped to provide the samples from the Bahamas and that went into my soft coral or octocoral work. And then Dr. Carrie Manfrino and her team of deep divers from the Cayman Islands provided the samples for the Mesophotic reefs. In addition to the folks that have helped me out with my academics, I have to thank my support structure, my family, my friends, and my lab mates. My parents, Jim and Mimi Crandall, my sister, Susanna Crandall, and my grandma, Dorothy, who are my biggest fans, my biggest cheerleaders, and always are pushing me forward. My friends here in Syracuse have been wonderful throughout this whole process. I'd like to particularly thank Sean for all of the perspective that she has given me over the past 2 years and all of the fun that we've had. I'd like to thank Casey for being a great roommate and a great commiserator as we both worked on the end game in our graduate careers. I want to thank Caress, who is by far the best lab mate and best roommate that anyone could ever ask for. I want to thank Ben, who was one of the first people that I ever met here at ESF and who showed me how to work with corals. And then I'd like to thank Lindsay, who has been through this process, has provided me with lots of good advice, and most recently has provided a roof over my head. Along with all of those folks, I want to thank the ESF community as a whole. I want to give a special thanks to all of the undergraduates who keep me on my toes and say hi to me as I walk around campus. I want to thank all of the faculty and staff that are always friendly and always willing to help me with what I need help with. So thank you very much.